So as you can see in this figure, pinnipeds, phocids, arose after cetaceans had already fully adapted to an aquatic lifestyle. This may mean that phocids and other pinnipeds have not yet had long enough to adapt to a fully aquatic lifestyle. Or perhaps, on the other side of things, pinnipeds have had the same amount of time to evolve to an aquatic lifestyle, and it has not yet evolved, so perhaps they are destined to remain semi-aquatic.
Due to the amphibious nature of phocids, they must use thermoregulatory traits to reduce heat loss and that are effective in both an aquatic or terrestrial environment. In general, specialisation for aquatic life has resulted in decreased reliance on external insulation such as dense fur and has been replaced by internal insulation through thick layers of blubber. Pinnipeds are the only mammalian lineage that has retained both types of insulation, and in phocids the use of different types of insulation can depend upon the maturity stage. Adult phocids reduce heat loss through smaller surface area to volume ratios than similar terrestrial animal like vasoconstriction, countercurrent heat exchange and insulation.
Unlike adult seals, most phocid seal pups are born with little or no blubber and rely instead on a fur coat. Phocid pup fur is lighter and more insulative in air than a similar thickness of blubber. Small-bodied animals can achieve greater insulation with less volume and weight using fur compared with blubber. However, when phocid fur becomes wet, water replaces the warm, trapped air in the underfur and heat can be readily conducted away from the body. Therefore, a fur coat is a good insulator for phocid species that live in dry environments or on stable substrates such as elephant seals and ribbon seals, but isn't as effective for species with high potential for early entry in water, which may occur in species born on unstable pack ice or phocid species that naturally enter the water early in life, such as hooded seals and harbour seals. As seen in this diagram, phocids, specifically hooded seals, have lost the regular elongated scale patterning on guard hairs and underhairs that are present in species that use fur for insulation in water, such as the fur seal and sea otter. This provides morphological evidence that both groups no longer utilise the fur for insulation in water. Among the phocids, phocines have significantly greater fur densities than monocenes, which may indicate initial changes associated with aquatic living that are reduced or lost in the later diverging species. These morphological changes observed in phocid fur provide evidence of convergent evolution associated with aquatic specialisation.
So adaptations for both phocids and cetaceans are similar in terms of the respiratory system. Oxygen stores in phocids are generally small lungs to reduce buoyancy and then also avoid decompression sickness. Surfactants in the lungs more so used for anti-adhesive functions, enabling the lungs to reopen after the collapse during deep diving. Blood volumes in seals are larger than quadrupeds, actually twice as much, and hematocrit value and hemoglobin content is very high, 60% compared to 45% in humans. Their oxygen carrying capacity in the blood is expected to be 45 milliliters of oxygen per kilogram in hooded seals compared to 11 in humans. A study discovered that there was an increase in hemoglobin concentration in diving Weddell seals that was released by large amounts of red blood cells from the spleen. Phocid muscles are often also very rich in myoglobin, being able to store around 6 times more oxygen in the muscles than humans. Oxygen stores in cetaceans are very similar, with the small lungs. The blood oxygen levels are around threefold the amount to terrestrial mammals. There seems to be a correlation between the deeper the diver and the smaller lung volume compared to their body size. Likely because the lungs are not primarily oxygen store for divers due to the negative effects of buoyancy and to combat the issue of decompression sickness.
Because phocids are semi-aquatic, their eyes and ears have to stay adapted to both underwater and land usage. The eye of a phocid is extremely well adapted to underwater visuals. In the graph on the left, a northern elephant seal only takes around 4 to 6 minutes to adapt to the same level of darkness than where a human would take 20 to 30 minutes. Both pinnipeds and cetaceans share similar characteristics of the eye. Their eyes have lost their S-cone cells, which is the ability to detect blue wavelengths. For hearing, the ear is neither specialized for underwater nor overwater hearing. The reason for this is because they need to attend to both airborne and underwater sounds, both of which are in very different media. This is in contrast to cetaceans, whose ear is very different as they can fully adapt to underwater acoustics. It seems that the pinniped ear is currently constrained during its evolution because of a semi-aquatic life. However, the phosid hearing system is the most adapted to aquatic environments of all pinnipeds, showing signs of losing quality of overwater hearing over underwater hearing. These changes are close to changes observed in the evolution of the cetacean ear. However, as always, further study is required to draw conclusions.
One characteristic of pinnipeds is the clear presence of vibrissae. Vibrissae are present in cetaceans as well, although they're not as defined and are absent in some species. There are different types and not yet understood if the different types serve different functions. Phocids have the most profound supraorbital vibrissae, or eyebrows, from all pinnipeds. They also possess rhinal or nasal vibrissae that is absent in other species. The vibrissae have been shown to be extremely sensitive, capable of detecting objects such as prey. There is also some evidence that the vibrissae aids in locomotion, creating laminar flow in a similar fashion as a humpback whale's flipper bumps. However, vibrissae remain largely understudied.
There is a lack of specialized biosonar system in phocids compared with cetaceans. Instead of detecting prey using sonar, they mainly use their highly developed eyes for seeing underwater. Pinniped echolocation is a highly debated topic. There is some evidence of seals using vocalization for detecting large objects and orientation, such as finding a hole in the ice cap. In the graph, we can see a series of sounds emitted by the seal getting more frequent the closer the seal is to an ice hole, and there is not enough evidence to draw conclusions on the functions of the vocalization. Interestingly though, it has been reported that seals in polar regions and ones that dive to extreme depths vocalize more than their other counterparts. There might be some evolutionary drive to develop an active biosonar system in conditions of near or total darkness. Only a few species exhibit the usage of echolocation, most notably odontocets and cave bats. Interestingly, these two species underwent parallel evolution. They separately mutated the same gene, Preston, that is a critical gene for echolocations, which phocids lack. Both of the echolocating species live in environments where visual feedback is poor or even non-existent, making echolocation very advantageous. There are obviously bat species that do not echolocate and do not have this gene, but it is hypothesized that the mutation of the gene led to a positive selection in these bats. Perhaps we can look at phocids that dive in extreme darkness the same way. If they do mutate this gene, it may be very advantageous and lead to the first echolocating pinniped species. What a time to be alive.
Evidence pinnipeds could go back. If we consider energetic costs, we see that they reflect different locomotor modes. In a study, Williams analysed the energetic challenges associated with changing from terrestrial to aquatic locomotion in primitive marine mammals and compared transport, maintenance and locomotor costs of extant mammals, varying in the degree of aquatic specialisation. Their research shows that running and swimming specialists have converged on an energetic optimum and are unlikely to show any evolutionary improvement. Plus, if we didn't consider the costs to maintain endothermy, their energetic costs would be similar to the ones of fishes, which display the lowest one. On the other hand, our pinnipeds' energetic costs are between 2.4 and 5.1 times higher than locomotor specialists. I think it could be easily hypothesized that they are a transitional stage. Evidence pinnipeds know what they're doing, they're not a transitional stage. Yes, such high energetic costs seem to go against the hypothesis that a natural selection maximizes fitness along the evolutionary pathway. However, I want to extend our research for an answer from locomotor and physiological mechanisms to environmental and ecological factors. We can presume that these provide semi-aquatic benefits that overcome the high costs. In line with this, a study on Weddell seals confirms that behaviour plays a key role in decreasing energetic costs for semi-aquatic animals. In particular, these seals exhibit high variability in their locomotory strategy while hunting, such as extending glides, burst and glide swimming, and roller coaster manoeuvres. Moreover, if we look at their fat-based metabolism as an adaptation to aquatic life, we find interesting researches. Unlike terrestrial mammals, the primary physiological role of leptin in marine mammals may not be an indicator of body fat. In fact, if leptin in marine mammals has contributed to regulation of fat deposits and energy balance, adaptive evolution of leptin should have occurred across all marine mammals, from cetacea to pinnipeds. However, this is not the case. Hammond revealed a substantially larger number of amino acids substitutions in harbour and grey seal leptin sequences than in any other mammals, and also that leptin in deep diving seals was expressed in the lung. They proposed a role of leptin in pulmonary surfactant production related to respiratory physiology, in addition to its common role in energy balance, confirming that the leptin genes of phocids have significantly diverged from other mammals. A further investigation on the diversification of leptin in phocids also provided evidence to support hypothesis of positive selection of the leptin gene within the phocid seals, but not within cetaceans, which supports the hypothesis that phocid leptin divergence is unlikely to have arisen by random mutation. Based upon these phylogenetic and structural assessments, and considering the comparative physiology and varying life histories among species, they postulated that the unique phocid diving behaviour has produced this selection pressure.
So, to conclude, adaptations in diving mammals are fairly similar across the different lineages. There is clear evidence to support the fact that phocids are not on an evolutionary track to being cetaceans. However, more research is needed to confirm this theory.